Today is we're going to continue from where we were yesterday. I'll start and I'll show you the blood flow through a pretty picture of the heart, and we'll follow the blood through those different chambers. And the next thing we're going to do is going to recapitulate the drawing we had on the board yesterday and finish the story on the different layers. We're going to talk about the endocardium, the fact that all those chambers and the valves are covered by those cells called the endothelial cells. Those endothelial cells will form the endocardium. And the endocardium then can be followed right out into all the blood vessels. So there'll be a continuous layer of those endothelial cells, and we'll walk our way through that. And then we're going to put on a new layer of cells on the outside of the heart. And that's going to form something known as the pericardium. Turns out that the heart sits in a pericardial sac. And the pericardial sac is composed of two layers of these so-called mesothelial cells. One layer of the mesothelial cells is attached to the wall of the heart. The other layer is attached to the body wall. That leaves a kind of opening between these two layers of mesothelial cells, and the opening region, the sac, the lumen of the sac, then will be filled with a little bit of fluid, not much, but a little bit of fluid, and that fluid provides a lubrication so that each time the heart fills with blood and then contracts, as it slides along the body wall, that movement, that sliding is lubricated by the fluid in the pericardial sac. But because of the anatomy, the way the pericardial sac surrounds the heart, there's a potential that the pericardial sac can fill with fluid if there's inflammation. That will bring us to the idea that for the myocardium, the muscle to work, we first have to fill the chambers. And it'll bring us to the idea that there is a low pressure return of blood from the vena cava, from the systemic vasculature, and from the pulmonary veins, from the lungs. That's a low pressure return. So there's not much force coming into the heart to expand the ventricles. So what happens is if this sac that surrounds the heart begins to fill with fluid, and the pressure inside that sac begins to become greater than the pressure of the fluid of the blood coming into the heart, then we can't move blood into the heart. And that's a really dangerous event. And we'll talk a little bit about how that happens and how that's an upshot of the fact that the pericardium is a closed sac and surrounds the heart. And then from there, we'll get into the cardiac cycle. Basically, the cardiac cycle is a series of events that repeats itself over and over and over throughout life. And those series of events begin with something known as diastole. Diastole is defined as the low pressure filling phase of the ventricles. So when you think about diastole, think about filling the ventricles with blood. Those chambers we talked about yesterday. Those chambers will fill with blood during this low pressure filling phase. Then we'll see the next phase will move from diastole filling to something known as systole. Systole is when the ventricular myocardium contracts. It's the contraction of the ventricular myocardium which generates force and applies that force to the closed volume of fluid in the ventricles. The blood as a fluid is non-compressible. So when systole begins, when the contraction begins, pressure builds and pressure builds and pressure builds. But that pressure will continue to build until that pressure is released or relieved by the opening of the aortic valve on the left side and the pulmonary valve on the right side. So we'll see then diastole begins. I'm sorry, let's do it again. Diastole begins with the filling of the chambers. Systole begins when the ventricular myocardium starts to contract. Systole comes to an end when the muscle is no longer contracting and the blood has been forced out from the chambers into those tubes, either the aorta or the pulmonary artery. That brings us to this cycle, and what we'll do here is we'll walk through the cycle verbally first, and then I'll draw a figure on the board and walk our way through it. But the cardiac cycle begins with diastole. There's a point called the end diastolic volume. At the end of diastole, when the ventricular chambers are filled, that volume in the chamber at the end of the filling phase is called the end diastolic volume. At the end of diastole, the pressure begins to build with the contraction of the ventricular cardiomyocytes. But because those valves, the aortic valves and the pulmonary valves, are so stiff and don't open easily, there's a long period of volumetric contraction. Isovolumetric means the volume is not changing. During isovolumetric contraction, the volume remains the same, but the pressure builds, the pressure increases. The pressure increases until such a time that the pressure is so great it pops those valves open. And now the only way for the blood to leave the chamber is out across the aortic valve on the left side and the pulmonary valve on the right side. And that means the blood will be ejected from the heart. So there'll be ejection phase. And then we come to a very brief period. This is only about 20 or 30 milliseconds. And it's called isovolumetric relaxation. Again, there's meaning in the term isovolumetric. The volume doesn't change. But what's happening is the muscle is relaxing. As the muscle relaxes then, and we no longer are generating pressure on the fluid in the ventricles, the isovolumetric relaxation now is going to be a brief period of time, and then we begin the process all over again, where diastole starts, and we allow the blood to come back into the heart, and we fill the ventricles with the blood in diastole. Then we'll finish with the pressure volume curve. We'll walk through this verbally, and then we'll put this on a graph, and we'll see what happens to the opening and the closing of the valves at each point in the pressure volume curve. But let me show you the picture first, and we'll walk through what we talked about yesterday, and we'll begin there. This is a pretty picture of what we talked about in the 4-box model yesterday. And again, to review it, in blue is the right side of the heart. So this will be deoxygenated blood coming into the heart. It's going to come through the vena cava, enter the right atrium, flow across the right atrioventricular valve, also known as the tricuspid valve, into the right ventricle. At the same time, blood is returning from the lung. It's returning to the left side of the heart from the lung through these two tubes, which are the pulmonary veins. Again, that's the terminology. Vein returning blood to the heart. It's bringing blood toward the heart. So the pulmonary veins are bringing oxygen from the lungs to the heart. And those pulmonary veins now empty into the left atrium. The left atrium then is now continuous with the left ventricle. These two chambers are continuous through this door, through this atrioventricular valve. Where on the left side, this is known as sometimes known as the mitral or bicuspid valve. The blood flows now in this direction. Then, at the end of the filling phase, once we've filled those chambers, then we'll begin to get contraction in the myocardium. The myocardial contraction, the shortening of the Z lines, the bringing together of the Z lines, will now generate force on the fluid in these chambers. The first thing that happens is that these atrioventricular valves, which are fairly floppy, those slam shut, blub. Then the pressure builds and pressure builds because these valves, the aortic valve, where's the aortic valve? The aortic valve up here, just the aortic valve. The aortic valve and the pulmonary valve are really pretty stiff, so they're much harder to open. So we don't open them until we've had some period of contraction of the myocardium, building pressure on the fluid inside the chamber, and that will then open these valves. Then the blood leaves during the ejection phase, and we start all over again. But that's the pattern we're going to talk about today. Yesterday we talked just about the flow. We're going to talk about the stepwise pattern today. But before we get to that, there are a couple of additions I want to make to the organization, the cellular organization of the heart. So I'm going to draw here. This is again the right atrium. The right atrium, the chamber is surrounded by atrial myocardium. The next chamber we talked about was the right ventricle. And the only pathway between those two chambers was across this right AV valve. The input to the right side of the heart is through this structure known as the vena cava. So the blood flow comes in through the vena cava, flows down across the open, let me open this up, down across the open right AV valve and into the ventricles. The pathway out is through this structure here on the right side, will be through the pulmonary artery. And right at the opening between the right chamber and the pulmonary artery is this pulmonary valve. The same pattern, pretty much, repeats itself on the left side. This is the left atrium, the left atriomyocardium. The input here is from the pulmonary veins. Again, the convention of blood being carried toward the heart in veins. And then the next structure here is the left ventricle. This opening in the middle, of course, is the chamber. And the pathway between the two is the left AV valve. The output here is into the aorta. And at the interface there is the aortic valve. We drew all that yesterday. And where we closed, and I won't belabor this too much, is that the chambers are all lined with a continuous layer of endothelial cells. That endothelium continues from the chambers over the valves and out into the blood vessels. So wherever we look in the cardiovascular system, there's this continuous layer of endothelial cells forming that lining, in the case of the heart, between the open chamber and the underlying muscle. In arteries and arterioles, we'll see the endothelium sits on a basal lamina and is separated from smooth muscle. But for our purposes here today, we have this continuous layer of endothelial cells. That lines the inside of the heart. Lines those chambers. But there's another layer of cells I want to tell you about here. And this layer of cells is on the outside of the heart. And I'm going to draw these in orange. These orange cells, we can give them a name. You can say that these are mesothelial cells. The mesothelial cells form linings over all of the organs in the body cavity. So as we work our way through this, we'll see a mesothelial cell layer in the heart and the lungs and into the gut. But what I want to emphasize today is that these mesothelial cells, which are attached by a basal lamina to the wall of the heart, form a continuous layer on the wall of the heart. But in addition to forming this layer of mesothelial cells on the wall of the heart, they fold back on themselves. I'll kind of have to show them in here. They go all the way over the atria. But they fold back, and they'll also then form a connection to another layer of mesothelial cells. And these are mesothelial cells attached to the body wall. So again, there'll be a basal lamina that will attach these mesothelial cells to the body wall. And what happens now if we close this loop is that these mesothelial cells form something known as the pericardial sac. So I think a plastic bag works pretty well to imagine what's going on here. So if these walls of the plastic bag are intended to be the layers of mesothelial cells, the heart sits down in that mesothelial cell layer, and the mesothelial cells then wrap up around the heart to form a kind of sac. And if we extend that analogy here, this side of the plastic bag is attached to the wall of the body. Oh, I'm sorry. This side of the mesothelial cell layer is attached to the heart. This side is attached to the body wall, but there's a space in between. This is just like there's an open space in the plastic bag, there's an open space here. So this is a fluid-filled space. Now that fluid contains some mucopolysaccharides, so it's slippery. And so that fluid now provides a kind of lubricant. Remember what's going to happen here. Day in and day out, 60, 70, or more times a minute, this heart is going to fill with blood. It's going to distend. It's going to enlarge. And then it's going to contract and it's get smaller. So you can imagine here how the wall of the heart is extending and then contracting, extending and contracting. Each time it has to move along the body wall, and so what the fluid in the pericardial sac does is it helps lubricate that movement, so that as with each contraction, there's a smooth movement between the wall of the heart and the body wall. So this mesothelial sac extends all the way around the heart. I won't do all of it, but note that this layout is the same as we've talked about for epithelial cells. So that there's an apical surface facing the lumen of the pericardial sac and a basilar surface attached to the wall of the heart. Again, over here, there's an apical surface that's pointing into the lumen of the pericardial sac and a basilar surface attached to the wall of the heart. It's these apical surfaces from which we secrete those fluids. So this is meant to be an apical surface. And that's where the secretion of fluids is into the fluid-filled space. There's two other pieces of nomenclature I should give you. This region of the mesothelium attached to the body wall the mesothelium that's attached to the body wall has a name. It's given a name. It's called the parietal pericardium. It's parietal because it's the body wall. But similarly, these mesothelial cells in here also get a name. These are called the visceral pericardium. The visceral pericardium is now that layer of mesothelial cells attached to the wall of the heart. It's attached to the viscera. And in this case, the viscera is the heart. Okay, so that's the anatomy. Let's spend a couple of minutes, though, emphasizing a term we're going to come to in a moment and why it's important in terms of this pericardial sac. One of the things I want to impress upon you is that the flow of blood from the lungs through the pulmonary vein, and the flow of blood from the systemic vasculature through the vena cava. This is a low pressure return. We're going to see a little later that the pressures generated by the contraction of the myocardium can on the left side reach well over 100 millimeters of mercury. But the pressures bringing the blood back into the heart are, for both the left and the right side, no more than about 3 to 5 millimeters of mercury. So this is a low pressure return. Now, one of the things I want you to sort of cotton on to here, because it's going to be emphasized as we go through the rest of the lecture, is that the cycle is going to be to fill the ventricles with blood, then contract the myocardium and force that blood out to the tubes. Do this over and over again. So the cycle is to bring the blood into the ventricles, both on the left and the right side, through this low pressure return. If you don't fill the chambers, you can't pump any blood out. This becomes important when we begin to think of problems, potential problems here with the pericardium. There is a condition known as pericarditis. This is an inflammation. This is inflammation of the pericardium. The upshot of this inflammation is often fluid buildup. In the pericardial sac. So you have to envision this pericardial sac as a closed chamber. Now we're putting more fluid into this closed chamber. There's no place for that. There's not much room for expansion. So if we put more fluid in here, what's going to happen to the pressure inside that sac? Put more fluid into a confined chamber, the pressures are going to go up. If this pressure goes up very far, beyond 3 to 5 millimeters of mercury, then the pressure out here is greater than the pressure in the ventricles that's bringing blood into that ventricle. So if you run into a situation where the pressure in the pericardial sac exceeds the pressure of the blood coming into the ventricles, you're not going to get any blood into those ventricles. That's really dangerous, because if you can't get blood into the ventricles, you can't pump it out. So this very delicate balance of these pressures is really important. Now, normally there's not an issue, but if you get pericarditis, and emergency room physicians are trained to see this. They're trained to watch for it. They're trained to listen to the heart and hear it. They're trained to observe it. And if they begin to sense there's a problem that the pericarditis is preventing the filling of the ventricles, they'll take this great long needle and a syringe, they'll poke it through your body wall, go right into the pericardial sac and withdraw that fluid. Because you have to reduce the pressure generated by that fluid right now. Because if you don't, and you don't get filling of the ventricles that can't pump blood out to the rest of the vasculature, death is coming pretty soon. So they're trained to watch that fluid and the pressure. And all of that simply to say, to emphasize the importance of this low pressure return, the low pressure movement of fluid into these ventricles. That's a low pressure return, which is so low that it can be exceeded by the pressure in the pericardial sac. Yes?
So this is kind of like what, a heart attack, you say?
So what's a heart attack? No, not exactly. This will cause a heart attack because it will prevent the flow of blood into the aorta and then into the heart muscle. But typically a heart attack, and something we'll talk about hopefully next time, is where there is an occlusion of the blood vessels that are going to the myocardium. This is a piece of meat. This is a big piece of muscle. And in addition to being a big piece of muscle, it requires lots and lots of oxygen. It's really rich in mitochondria. And if something happens that we block one of the blood vessels, the major blood vessels that feeds oxygen to this myocardium, then we have a heart attack because we can't deliver adequate oxygen to those cardiomyocytes, those cardiac muscle cells, to keep them alive, and they die, and that's a heart attack. It usually happens when we do something to occlude those vessels. Usually there's an embolism, a clot. Sometimes we'll peel off a piece of plaque that's built up in those coronary arteries and it plugs those vessels. We lose oxygen supply to that portion of the heart. Those muscles die. They can't regenerate. That's the damage from a heart attack. Anything else? Okay, so that's the anatomy. Oh, yes, I'm sorry.
Are the left chambers connected to the right chambers at all? Like, is sharing blood or pressure at all? Like, I know that
Is there any, so the question is a good question. Is there any sharing between the left and the right chambers? Not in the adult. If you get into the details of what's happening in the fetus, it's different. Because in the fetus, you don't have to worry about pumping blood from the right heart to the lungs. All your oxygen is coming from mom. And so there you will have, at least early on, there will be some openings of the chambers. But in the adult, there are no openings. This is a separate chamber from this chamber. And unless there's a mistake someplace, there's no mixing of that blood. There are some birth defects where the blood does mix, but it shouldn't. Yes? Did you have a question? No, okay.
Are the endothelial cells different from the mesothelial cells just in the fact that they are on the inside of the heart rather than on the outside?
So what distinguishes the endothelial cells from the epithelial cells from the mesothelial cells? Fair question. And it's a little nebulous, but the way I distinguish them is if it's an epithelium to me, that you must be able to follow that cell layer to the outside world, epi upon. So we're going to talk about an epithelium in the pancreas. It's going to be basically an exocrine gland. So we can follow those into the epithelial cells of the lining of the intestine all the way to the outside world. It's a proper epithelium. When we talk about an endothelium, it never sees the outside world. It's confined to the cardiovascular system. The mesothelium is in between. They're a separate set of cells. Think they're separate embryological origin. And these mesothelial cells now are confined to forming the linings over the surfaces of the organs in the thoracic and abdominal cavities. So those are the three separate cell types. If you look at the way they're organized, they sit on basal lamina, they're very similar. If you look at what they do, they're in many cases very similar. They have apical and basilar surfaces, but because they are linings in different places, we give them different names. Yes?
Just to clarify, so the pericardial sac is just to surround the heart and protect it, right?
So the question, what does the pericardial sac do? To my mind, it's most effective in that it provides this slippery lumen so that when the heart beats, when blood comes in and expands and then contracts, because this is going to be moving throughout life, this reduces the friction of movement of the heart's wall with respect to the body wall. So it's basically a kind of buffer where we can easily slip one by the other. That's its main function. Yes?
My question was, so for the mesothelial cells on the like along the heart, the cavity of the heart, and the mesothelial cells onto the body wall, where do they connect? And is the ones connected to the heart connecting like both sides of the right ventricle and the right?
So the mesothelial, see if I can answer this. The mesothelial cells, one set called the visceral mesothelial cells, are attached to the wall of the heart. So there'll be a layer that goes over the atria too, as far as I know. Then someplace out here, this layer of mesothelial cells attached to the wall of the heart then forms a new layer. It's continuous. So you could get on one of these cells and walk along it. And if you could see through the mesothelium, you'd see the wall of the heart. You'd come out here someplace, you'd leave the wall of the heart, you'd look one way and see the lumen, the other way, then eventually you'd come around and you'd look at the body wall. So it becomes a continuous sac. So Where it begins and ends, I'm not sure, but I think what's happening during development is those mesothelial cells are attaching on this side to the body wall of the heart, on this side to the wall of the heart, and on this side attaching to the body wall. But someplace, you're right, they must make that loop, but I don't know exactly where that loop is made. But it does form a closed sac, that I'm sure of. Anything else? Yes?
How does it, if it forms a closed sac, how does it form around the aorta and the pulmonary artery and all of those?
My understanding is that it will attach through its basal lamina to those structures. They extend over the atria, and then will attach to the body wall. And that will be a continuous layer. And then you have the whole segments attached to the body wall. So you can imagine the apical surfaces, one facing the lumen, where you have let me try it again. You have the series of mesothelial cells attached to the wall of the heart. Then you look at the apical surface, that's facing the lumen. Someplace, and I'm not sure where it happens, but someplace the visceral pericardium then becomes continuous with the parietal pericardium. And that forms a closed sac. And the parietal pericardium is now attached to the body wall. Okay, now exactly where that happens, I'm not sure. Someone knows. But I'm not sure exactly where you move, where if you would stand on that pericardium, where you'd be able to see the junction between that which is visceral pericardium and that which becomes parietal pericardium. There's a place in there, but I don't know where it is. Okay. So that's the anatomy story. And that gives you some sense of the low pressure return. But now what we want to do is we want to begin on this board talking about the cardiac cycle. Now it's a cycle, so we have to break into it someplace. And so typically the way we break into the cycle is we label the first one as diastole. Diastole is the period of ventricular chamber filling. With blood. Another way of saying this is that diastolic filling is when those chambers are using the low pressure return of blood to fill. So think of diastole as the filling phase of the chambers. The next concept is the concept of systole. Systole occurs when we have myocardial contraction. When the Z lines are shortening during systole, pressure increases on the blood in the ventricular chambers. If we go back up to that upper panel, you can imagine what we've been talking about all along, where blood is flowing through the pulmonary veins and through the vena cava in that low pressure filling phase. It's flowing across the open atrioventricular valves and is basically filling these ventricles. That's all diastole. When you're filling the ventricles with the low pressure return, that's diastole. But we have to think about how we're going to propel this blood on the left side out of the chamber into the aorta and to the systemic vasculature, and on the right side out of the chamber into the pulmonary artery and through the pulmonary vasculature. How are we going to manage to move that blood? The movement of the blood occurs here with systole. The force that's generated to move the blood is the force generated by the shortening of the sarcomeres during muscular contraction. Then systole has a phase during it called the ejection phase. The ejection phase is when blood is ejected from the chambers. Across the pulmonary and aortic valves. So if we come back here again, diastole is when the blood comes in. We come to the end of diastole, we've filled the chambers. We'll see at the very end of diastole there's going to be contraction of the atrial myocardium that will give the final squirt of blood from the atria into the ventricles. But when the filling phase ends, when we come to the end of diastole, then we begin to invoke contraction of the myocardium. When we get the myocardium to contract, when these Z-lines shorten, by shortening the Z-lines in a closed container, because remember, when this begins to shorten, the first things that will happen is those AV valves are going to close. So if things are working properly, we can't move any blood from the ventricular chamber back into the atrial chamber. So we snap these valves shut. But if you've ever listened to someone's heart, you know there's a period in between. You hear the lub sound, then you hear the dub sound. That first sound is closing of these AV valves. If you have a stethoscope, sometimes you can actually hear it. You can hear when this valve closes, then there's a delay and the pressure builds, the pressure builds, the pressure builds, and then you can hear the opening of these valves as a swish of blood out into the aorta and a swish of blood out into the pulmonary artery. That swish occurs after the valves have opened. But there's a delay between the time we fill the ventricles and the contraction builds and builds and builds and builds and pressure builds and builds and builds until there's enough pressure inside the chamber that we snap these valves open. When the valves open, there's the ejection phase, blood leaves, but then the contraction of the ventricular myocardium stops. We stop sending signals to that muscle. We no longer get contraction. When it stops contracting, then pressure inside the chamber begins to drop. Because the only way we generate pressure is with contraction of the muscle. But if we're not contracting the muscle, there won't be any pressure. If we don't have any pressure in here, we have lower pressure in here, there will be pressure out here remaining both in the aorta and the pulmonary artery, and those pressures now will snap these valves shut. That's the dub sound. That's the second sound. And we snap those shut. And then we start the cycle all over again. Yes?
So would like a heart murmur be the valve?
So what's a heart murmur? It's a great question. There are a number of different heart murmurs. The most common is when these AV valves don't shut properly. Could be on the left side, could be on the right side. And what they hear in a murmur is that when the muscle contraction begins, because these valves aren't closed properly, you're getting what's known as regurgitation. You're getting fluid, blood, moving back from the ventricular chamber into the atrial chamber. That's the murmur. Now, sometimes there'll be problems with either the aortic valve or the pulmonary valve, and you'll also hear murmurs then, too, because when this contracts, and then the valve is meant to snap shut, if it doesn't close properly, there'll be murmur of the fluid, the blood moving from the aorta back into the ventricle. That's another kind of murmur. So if someone tells you there's a murmur, you need to ask which valve, because it could be any of them. And sometimes you'll see something known as stenosis, and particularly in the aortic valve, and that's when they don't open enough. So they're kind of closed shut. And you'll often, and physicians can hear that too, because it's not a nice wide opening, it's a narrow opening. It's like when the Poudre River comes through a narrowing of the canyon. You have to move blood through here faster, and so you can hear it. And so on auscultation, you can hear what they call a stenosis of these aortic and pulmonary valves. So people who are trained to hear this stuff can hear a whole bunch. I can't. Yes.
So when you have that heart murmur and the valve doesn't open or close properly, is that when they go in and replace the valve?
That's when they go in and replace the valve. There are some cases, somebody I know actually, had a faulty tricuspid valve when he was born. He didn't have all 3 leads, he only had 2. He didn't know it for a long time. It wasn't until he was like in his 50s or 60s that he began to have problems. They diagnosed him with a faulty leaf on his tricuspid valve, and they went in and replaced it. He's fine. We've gotten pretty good at this sort of thing. 50 years ago, not so much, but now we're pretty good at it. Anything else? Okay. So we've gone through this where we have filled the heart chambers, then we have increased the pressure, we forced the blood out into the tubes in this ejection phase, and the third thing I'm going to tell you about is a very short period. It's only about 20 to maybe 30 milliseconds long, maybe less. And this is a period known as isovolumetric relaxation. Now, this gets to be kind of nebulous, and the question always comes up, well, where does systole end and where does diastole begin? Well, if diastole is defined as the filling phase, when blood is returning and coming into those chambers, well, that's not happening here. This is isovolumetric. So this really isn't part of diastole, because diastole is defined as the filling phase. And yet, systole is defined as the contraction of the ventricular myocardium generating pressure. Well, by the time we get to this number 3 point with isovolumetric relaxation, the myocardium isn't contracting. So it really isn't in systole. So to my mind, this period of isovolumetric relaxation is kind of in that netherworld. It's neither systole nor diastole. It's a brief period in between. But these are the cycles. Blood comes in during diastole, the muscle contracts, the muscle generates force, it forces the blood out of the chambers, all of that systole, and then we have a period of isovolumetric relaxation, and we start over again. But I want to emphasize this period of isovolumetric relaxation for just a moment. Because I want to think about what's gone on here in the myocardium. Now, this is not going to be at all to scale, but if we looked at the myocardium, we can imagine the myocardium muscle cells as being typical striated muscle. And what I'm trying to draw here are the sarcomeres. So this is meant to be a sarcomere. And we know from studying skeletal muscle that when the muscle contracts, those Z lines get closer together. That's going to happen here too. So when we activate the ventricular myocardium, we're going to shorten the distance between the Z lines. We're going to get it to contract. That's how we generate pressure. That's how we generate force to drive the blood out of the right and left sides of the heart. But we've drawn the Z lines together now. How do we return them to their original length? Remember, we had that problem in skeletal muscle too, but we solved that pretty easily because we said in skeletal muscle, if I imagine the sarcomeres in my biceps and I pull my hand up to my arm, I've contracted those, I've shortened those sarcomeres by contracting the muscle. Now, in order to reset the sarcomere length of my bicep, I need to take my tricep and extend my arm out again. So what we we use these antagonistic muscles to reset sarcomeres after a contraction. But we don't have any such arrangement here. There's no antagonistic muscle. So we have to think about what force we're going to use to reset the sarcomere lengths. It's pretty straightforward. What we're going to use is the low pressure return, that 3 to 5 millimeters of mercury, returning the blood from the systemic and pulmonary vasculature into the ventricles, and we're simply going to use that low pressure to distend the ventricle. We're going to reset these sarcomere lengths with the low pressure return. That resets the sarcomere length. We can begin the cycle all over again. But it's the diastolic filling phase that's resetting the sarcomere lengths. So let's take this pattern of diastole, systole, and isovolumetric relaxation, put it into a graph. And I'll do that over here. This graph is said to be the pressure, volume curve of the cardiac cycle. It's often referred to as the toast diagram, and you'll see why when I draw it. Let me put this in the backboard so I have some room. So when we plot this is going to be an unusual graph. There's no time in this graph. This is volume on the axis, and this is pressure on the ordinate in millimeters of mercury. I'm going to start down here, and I'm going to say this is point A. Point A, when we look at the volume, this is typically 50 milliliters. This 50 milliliters at point A gets a name. It's said to be the end systolic volume. This is the volume of fluid in the ventricles at the end of the cycle. And we have to start someplace, so we'll start here at A. Then we have this period here that goes from 50 to, about 120. And if we follow this, it's a straight line for quite some time. And at the last minute, it goes up a little bit, and we arrive here at point B. What we're looking at between point A and B, let me put it out here, A to B, is diastolic filling. So between point A and point B, the volume's increasing. It's increasing from 50 mils to 120 mils. And this point out here, at the end of diastolic filling, we have this point here said to be the end diastolic volume. Typically, in resting heart, it's about 120 mils for most of it. It can be more in some people, and we'll talk about that. But typically, the end diastolic volume is about 120 mils. What this represents is this represents the volume in the ventricles at the end of diastole. But look at this curve. Spend a moment with this curve. If this is pressure in the ordinate, what's this little bump telling us? It's telling us there's an increase in pressure. When is it occurring? Well, it's occurring right at the end of diastole. So where do you think that increase in pressure is coming from? It's coming from the myocardial contraction of the atrium. Because at the very end of diastolic filling, this very thin layer of atrial myocardium contracts. It's not a very strong muscle. It doesn't produce a whole lot of pressure. But what it does is it produces the final squirt of blood across the AV valve and into the ventricle. That's the end of diastole, when we contract the atrial myocardium. And that's shown on this graph by this increase in pressure. Now, from point B to point C, This is said to be the period of isovolumetric contraction. There's meaning in that term, and I have to finish here shortly. This is the isovolumetric contraction. It's telling us the volume's not changing. This volume goes straight up. This line goes straight up, so the volume doesn't change. But then at C, the valves open. We get the ejection phase. We come to point B, come back down again. And this, CD, between D and A is said to be the period of isovolumetric relaxation. So this curve, this pressure volume curve, this TOAST diagram, tells us everything we said in the upper panel. And your job between now and tomorrow will be to study this curve and figure out where all the valves are opening and where all the valves are closing. If you understand the opening and closing of valves on this curve, where pressure is increasing and decreasing, you understand the cardiac cycle. Yes?
So D to A is the relaxation phase?
D to A is isovolumetric relaxation. You notice there's no change in volume.